Hi, everybody. Today we're talking about scientific writing. My name is Sina Bartfeld. I'm a professor for Medical Biotechnology at the Technical University Berlin, and I've been asked by my students to help them with scientific writing. This goes particularly to beginners, people that have to write the bachelor thesis or maybe the master thesis, but the principles here are really applicable to all papers as well. And I only learned them kind of by two scientific writers, Leslie Ogilvy and Rike Zietlow, both scientific writers that I had the pleasure to work with when I was a PhD student. So it's kind of more learning by doing, and I haven't studied this or so. And I think if you're a beginner, it's really hard work and feels like a big task. And now in hindsight, I realize very much how much these very simple rules, they're really true and they're very helpful in the process of writing. And I wish I would have understood them a bit earlier and practiced them a bit earlier and practiced them a bit more. So this is kind of just try to put it up to you guys so your journey will be a little bit easier than I found it. All right, so we will talk about kind of what rules exist in scientific writing, how you write your aims, your results and figures, discussion and introduction, material and methods, and abstract and practical advice. There's also by the publisher Elsevier a really nice online course. It's for free. You can register, and I can highly recommend that. So I found that very helpful. And obviously, what I tell you now is clearly inspired by Leslie Ogilvy and Rike Zietlow, and I had Leslie's old slides as a help, so that was really good. So let's start. So first, why all the rules? Well, I found it really, really hard when I had really no instruction. I basically had, so this is examples from others, just write your thesis similar to that. And that's a lot how it's done, but there are a lot of these rules that are kind of implicated. And I think nowadays you learn a lot to discriminate between an observation and the interpretation of the observation, and that one belongs to the results, the interpretation belongs to the discussion. This, I feel that it's nicely discriminated, but there's lots of more rules like, okay, figures should be self-explanatory, but what exactly is meant by this? And all of these rules or these guidances are meant to make your written piece, your paper, your thesis, easier to read, easier to understand. You want the reader to follow. And this is a parallel with all writing procedures. And there is a journalist teacher that used to say, basically, one person has to do the work, and it's either the writer or the reader. So you have to work on these things over and over again. And I just talked with my husband about it, and he said, it's kind of like when you're ice skating, when you're a professional ice skater, it looks like it is all so super beautiful and simple, and it's meant to look easy. And it's a bit same with writing. Writing is meant to just flow into your mind. The reading is meant to flow into your mind, make you easily and intuitively understand what was done, etc. But to get there, the writing procedure is actual work, like a training for ice skating, like any kind of thing that you get good at. You have to do work, and that can be difficult. Okay. The structure of your paper is really the structure of any paper. You start with your results. That's the main thing. You need kind of the building blocks of your paper are your results. That's the data that you have, the data you acquired, and that you present in figures. That's the main part of your paper and the most important. These results you then can discuss, right? Because you have to give them interpretation, you have to give them meaning. And this interpretation and meaning comes, of course, only really if you embed them in the current literature, if you shine light on them from all the different perspectives. What is known? What did other labs do? What else can be done? And this is done also within the discussion. You embed your own results within the current knowledge. Then once you know what are my results and what are the discussion, you also know what you actually have to know to understand the results in the discussion. That's the things that you need to introduce. And your introduction is kind of like a funnel. It comes, you pick up the reader at an interesting point where you think you can kind of pick everybody up with something like that's what's where the important comes from. For example, this disease that you're talking about will kill so many thousand people, whatever, something like this. And you pick up the reader with this, and then you explain more and more of the building blocks that you need. And they're like a funnel. They're a one-way street that point to one particular gap of knowledge. And this gap of knowledge, you have to spell it out. You have to indicate all this is known, but what is still only understood fragmentally is this or that, right? So we understand all this, but knowledge of this remains fragmented. So you indicate the gap of knowledge. From this gap of knowledge follows your aims directly. What do you aim to achieve in this thesis? Okay? We will go through these parts now in the lecture and see what is the structure of each of these parts and how can we use this structure to know how to write them. So the structure of your paper is really the structure of any paper. We will try to, that's why it makes a lot of sense to actually read a lot of papers and try to analyze these papers and try to find the structure that we're talking about in other papers. When we do this in the lecture hall, we actually take out example papers that we defined beforehand and try to find the knowledge gap in these example papers. But you can take any other paper of your choice. Just to point you to what I mean, we will take an example paper that happens to be the last paper from my lab. It's Nature Communications, published in 2022, and the title is Helicobacter pylori shows tropism to gastric differentiated pit cells dependent on urea chemotaxis. So let's go into this. Where would I find the knowledge gap? I just mentioned you start, the introduction, which is in a paper. It starts here. So here you have the abstract, then you go for the introduction, and somewhere here, usually in a paper, comes the knowledge gap. And then in a paper, there often comes this region of here we did this and that. So let's look at this region here a bit more closely. Okay, so here, this is the sentence it starts. In vitro studies in adult stem cell-derived organoids and organoid-derived monolayers have demonstrated that gland cells mount a strong inflammatory response, while the pit cells are relatively refractory to infection. However, it remains unclear whether Helicobacter has a preference to attach to and translocate CAG-A into a particular cell type. Okay, this is the knowledge gap. It's clearly stated. It always goes with these sentences. However, it remains unclear, or knowledge of this remains fragmented, or some sentence that states specifically what is not yet known. And then from this follows, briefly under that, what we do here, the aim. Yeah. Here we combine the power of human adult stem cell-derived gastric organoids and single RNA sequencing to identify the Helicobacter preferential target cell. I have, in this paper, we sort of inserted a bit of a technical paragraph to explain now that this fantastic technology of single-cell RNA sequencing is now available that would enable us to actually address this question. It's not necessary. You can also go directly from your aim, directly from the knowledge gap to your aim. Now, there's a difference between how the knowledge gap and aim follows in a paper versus a thesis. In a paper, it's like this. It usually goes knowledge gap, and then here we do this and that. In a thesis, I really like to have a separate section, aims of this thesis, which means that you come after the introduction with a separate heading, aims of this thesis, something that briefly states again the overall problem, gap of knowledge, and then specifically states the aim of this thesis is this and that. You can also go, my hypothesis is so and so, the aim of this thesis is this and that. For this, I will do exactly objective 1, objective 2, objective 3. Yeah, you can break these down into smaller chunks of tasks. The difference in formulating this is in a paper is here we did this and that. In a thesis, my aim for this thesis is, so it's a bit directed to the future. Okay, but the principle, identification of a knowledge gap, naming the knowledge gap, and then following from what needs to be done, this is the same. So how do you write your own aim? Well, avoid sort of very, very broad questions, something like every biological question is fascinating, or also avoid a sort of stepwise description without reasons for it, why this is done. So something like, then we did this, then we did this. The first focus was here, the second focus was there. It may be that you did all this, and it may be that it makes a lot of sense, but it is more intuitively easy for me to understand it if you tell me the flow of logic. Okay? It is difficult for me if you tell me lengthy details, especially of methods. You can specifically name some methods, but not in very long detail. Instead, it is helpful if you name specifically your importance, your hypothesis, your aim, and you maybe can break down those aims into smaller objectives. Be very concise and precise. I will show you some examples. This here is one study that I had difficulties in reading. You see that the aim of the study is relatively long. It starts with the sentence, every pathogen that affects the well-being of human is important to be investigated. So it starts very, very broad. It's unspecific. And this person has then also kind of got a bit into also it names relatively many citations here and details, and then it goes in one focus and then the next focus of this project. So it's difficult. Information is in there, and the thesis, the practical part of it was really good, but it was just not very well digested to make it easily understand for the reader. Another example here is this one. It is the absolute opposite. This is the minimal, super concise. This person has started with telling us where this person did his or her thesis. This does not belong to the aims. But then this person has clearly stated, we hypothesize that hematopoietic cells, particular monocytes, can be reprogrammed into this using these factors. The main aims of this study were 1, 2, 3. Very clear. And the third one that I would find the preferred format is a mixture of the two. It is exactly 1 page long. And this person starts with the specific yet relatively broad problem. The balance of stem cell proliferation and daughter cell differentiation is tightly regulated in the gastric epithelium. An infection with pathogens is likely impacting this balance by targeting subpopulation of cells, inducing cellular damage, apoptosis, and subsequent proliferation. Now comes already the gap of knowledge. Specific proteins mark stem cells and differentiated cells, but the function of many of these proteins remain to be elucidated. Then this person introduces the two main proteins that this person works on and says clearly, in this thesis, I aim to better characterize this protein expression and this protein function in the gastric epithelium. The specific aims are, and then comes the list of aims, broken down into a bit more methodological objectives. Okay, so you have the aims, 3 aims broken down into objectives. This I find is a good mixture of a bit of an introductory overview, which topic are we on, why is this important, what is specifically the knowledge gap, and what do I do. This is helpful for the reader because it just summarizes the introduction, puts you down on the spot to the point before entering the result. Okay, now how to formulate your particular aim. That's really hard and it takes practice. And actually your aim may shift during new data coming in, and then things change because it's real science. But how do you define your aim? You can just try it. Try to state in a short sentence, my hypothesis is, the aim for my thesis is, to this end, I will follow the 3 main objectives, or 3 main tasks, or however you want to say. I do this a lot. I do this with colleagues. It helps me very much to do this with people that are not really related to this project, and maybe that don't even work in biological sciences. I like doing this with my husband because he really knows what I'm doing by now and he understands if I explain, but he will not be so super interested in the nitty-gritty of some pathway details or so, at least if it's not his topic. But if it's my topic, it's kind of like, yeah, okay, but get to the real shit. Tell me why this is important. And this makes me step away from these details and focus on, okay, what is the elevator pitch? What is the main message that I think is important and I have to tell? That is the name, the aim, the hypothesis, and the object. So if this is hard, practice this with anybody that doesn't run away fast enough. Try it on your roommates, your parents, whatever. By trying to do this, it helps you to sharpen saying exactly and concise what you want to say. Also, it can happen that there is a gap between what you originally set out to do and then what is actually put into the paper. In the paper, you tell a story. This means that you don't necessarily tell it in the order that it really happened. Okay? So your first task for writing is now define your message. What is your story? What is the problem you will solve? And why is this important? Discuss your data with colleagues and your supervisor, obviously, most important. And provide one simple and powerful message. And if you have too many stories to tell, then you're not there yet. If you write a paper on too many stories, it will be rejected because one paper is about one story. And even if you have the most beautiful wealth of data and it does not fit to your story, throw it out. It's not part of your story. Why? Because it is hard to follow and people have difficulties in reading it and concentrating it. So you need to tell them one story at a time. Focus. We do this many, many times for each paper. It's sometimes very painful, but I actually enjoy the process. It's kind of more carving out the real story. And often many stories you can tell from different angles. And it's kind of nice to play with that. So the results section. Clearly you describe your data. That is fairly obviously because you are in the results section. But how do you get there? Well, what we try to do is we try to first state short sentences of what we think we achieve to show. Each sentence should then make one figure. One example in the paper that I will use throughout this lecture, Aguilar et al., figure 1, the statement is there, organoids and organoid-derived monolayers represent different regions of the gastric unit. How do we get where? Well, I literally take a large piece of paper and write those sentences on it, and I draw the results that we have kind of fitting to, okay, to make this statement, I need this piece of data here and this piece of data here, and there we may still have a gap. So what would be the experiment that is still needed here? And then next sentence would be this, and next sentence. So I have the story on one piece of paper that actually hangs on my wall for some time because we work on this and this paper gets exchanged. But you have to start writing these statements down and trying them on. It's like putting on some clothes, see whether they fit, see whether they look good, see how the team reacts if you discuss this. Yeah. These short statements often become the figure legends, but not necessarily. This is a matter of style, and there's many people that dislike these statements. So what I mean with these statements is, for example, this statement, yeah, organoids represent different regions of the gastric unit, versus it could be, so this particular figure could have also been said, RNA single cell sequencing of organoids versus organoid-derived monolayers. Stop. So this is one sentence states the result, and the other figure legend would rather name what you did or name the technology. I personally find it, again, easier to read, easier to follow if I hear the statement, because then I know what you want to show me. And then I can evaluate whether I can see the same thing, or maybe I have critics or whatever. But if you tell me in the figure legend what this figure shall tell me, then it's easier for me to read simply. Okay, but it is a matter of style. It's not only this, and this is not the only one. Let's look at one figure. So here we take figure 4. Doesn't matter. We can take any figure. In any figure, here I chose from a paper a multi-panel figure, which means that a lot of data is put within one figure. All this data supports the same point, the same statement, the same sentence. And within each figure, these sub-figures make a logical flow. You could, however, easily take each of these sub-figures and make them a separate figure. Or you can also, actually for this figure, it would make a lot of sense to just take A, B, and C being one figure, and this here being the description of how we did the experiment for this. And then in the next figure, these could be one figure together, etc. Okay, but we need a logical flow within within one figure. Now, within one figure, you really need to define all the symbols and abbreviations and follow nomenclature rules, which means that you write down the gene names following the nomenclature. If you want to know which gene name is the right gene name, you go to the NCBI gene database. And there you look up the primary symbol and you use that symbol. If you talk about a protein, it is not italics. If you talk about a gene, it's in italics. That's why here I can already see, just by the way how this is written, that we're talking protein here. And this here is clearly a Western blot. I have the kilodaltons here. Here I have the sizes, and this indicates which band this is, because of the antibody stain. Now here we put a lot of thoughts on how to actually label the different conditions that we show here. Here are organoids within one particular growth factor condition. Here's a different condition, and here's the third condition. So we compare 3 different growth factor conditions. And we put a lot of thought into how to display them. And we use the same color coding throughout the figure. And we kind of introduce the color coding here, by here spelling out each of the abbreviations. Yeah, so this is the standard medium with EGF, noggin, arspondin, Wind, FGF10, gastrin, and TGF-beta inhibitor. In short, ENRWFGTI. If I now add nicotinamide, I have another NE at the end. If I don't have this nicotinamide, I have exactly these abbreviations. And if I remove Wind, then the Wind here in the media here is missing. And now having this defined, which is an experimental detail, but it's the important experimental detail for this experiment. What are these 3 conditions different in? And then if I can simply look at this, I see these are the 3 different conditions. And I see here, okay, if I add nicotinamide to the medium, obviously all MUC5AC is gone. If I don't add nicotinamide to the medium, but I just have the standard medium, I have a little bit MUC5AC. And if I remove Wind, I have a lot of MUC5. So this is what is meant with define all symbols and abbreviations, the important ones. And I know that's super hard in the beginning to know which abbreviations shall I introduce and which not. Gene names, you don't need to introduce because you have to introduce them into you have an abbreviations list and you introduce these gene names within a text. But if you have one particular condition like we would have here, this needs introduction. Otherwise, the reader would just get here funny symbols and would have to go to the text to understand what each of these symbols mean. This is meant by trying to make a figure self-explanatory. When I write a paper, I try, not perfect as this, but what I always aim for and try to is if I give this to a colleague, that this colleague would be able to see this figure only and kind of grasp what this is about and what I mean with which. Okay. Avoid lengthy description of methods, obviously, and put a graphics instead. As you see here, I like this very much. This here is the culture conditions that explain the experiment for B. Now, having defined these culture conditions, I now do my actual experiment, adding Helicobacter on this. And if I now add Helicobacter on this, I then look into how many bacteria bind to the cells in this particular condition. Again, same conditions that I learned already in the color coding. And now I see here as well that my Helicobacter binding here seems to be very low in this orange condition, higher in this medium condition, and even higher in the green condition. But is this statistically significant? I have to do statistics on this, and you have to do statistics on biological independent experiments. That's why we use different patients, which I indicate here. And each of these patients have different symbols that if you would look very deeply into this, you could see that. But then if we know, okay, this is different patient material, we can do statistics on those. And then I actually write down here the p-value. Some people like putting stars and then defining the stars in the legends. Many journals accept this as well. There's also journals that don't like that, and I personally don't like it as well. I like to see the p-value. Because then, be precise, tells me exactly. Figure gets a little bit busy there, I admit. You have problems with the font size and putting all the information in there. But you need to define every little thing here. So this here is then Helicobacter positive cells in percentages, and this is colony forming units, how many per well, and this here is the mRNA expression of the bacterial CAG-A. This is why it's in italics, in fold over. And fold over what? Okay, these ones are all put to 1. So our intermediate condition here is obviously set as control. This I can only see because it's put to 1. Okay, that's just as an example. This is arguably, you can probably do this even better. This is just a way to try to put as much information as a potential reader would need. Define any sizes, bars, error bars, etc. So if we put anything in here, we need to define this in the figure legends. Very typical is if you show if you show something, so I'm fairly sure that somewhere, if we show this bar here somewhere, it will say that this here is a median or a mean. Okay. Any microscopy images needs to have a size bar. The size bar is defined also. Include the number of replicates, n. A technical replicate. So imagine you do a qPCR and you will pipette the qPCR in 3 wells. You may have 3 results from your qPCR, but the original DNA that you did this on, so the cDNA that you did this on, came from the same RNA. So this is actually one biological experiment that you have put into technical replicates for the PCR. So these technical replicates control the PCR, how accurately you pipette the PCR, but not the biological experiment. The biological experiment can only be controlled by having several independent biological experiments. What are biological independent experiments? Either you use biologically different individuals, meaning different patients, like it's here in this experiment. Other have mice or something else. If you work with cell lines and you simply don't have biological replicates, then you can use independent experiments, so that you see the cells in completely different weeks, that these cells are within a different experiment. And you can only do statistics on independent biological experiments, a minimum of 3, and look up which test is actually allowed for which kind of data. And then within your figure legends, you need to state what is your N. We actually somewhere, yeah. So we define this here fairly clearly here. So here the statistical analysis was performed using one-way ANOVA with this and that comparison, etc. The data from organoid lines derived from individual donors are shown as symbols. Patient, this shows this and that, with the mean values with horizontal lines. Data in panel this and this are representative of at least organoid lines from 4 donors, and these are 3 donors, and this is done in how many experiments. So we actually spend some lines in defining exactly what is shown in the data. That's why our legends become a little bit long. This gives you all the information to really evaluate how robust the data shown is. Okay, and now I will show some examples from master thesis that I didn't do, obviously. And by showing somebody else's results, I wanted to make sure that you understand that I don't want to pinpoint to somebody that has done a particularly good job or somebody that has done a particularly bad job. But I think that by showing examples and making you go through the figures with me, you realize that one figure may be harder to understand and the other figure may be easier to understand. And this, I hope, would help you to define more of which information do I need to put that makes the reader being able to read this. So don't take this as a finger pointing, but rather, I hope, as a helpful example. Okay, so this is an example from a master thesis that I found understandable, good. This person has shown here a graphical overview. This is about some plasmids putting into an electroporator and then showing me the gene sequence that is targeted, and that here one positive clone is it comes out of this electroporation before it was selected, and after the selection there's a lot of these positive clones, and then showing us some more detailed images of these clones, and here also in another technology, in fact demonstrating that there is a positive GFP particular. So I can immediately understand what is this about. Several things are put together to make one point. Point is also clearly stated. I see axes being labeled. I see scale bars being put in many, not all of the figure and not all of the subfigures. But overall, I can get what this person is trying to do. This is another good example. It's maybe a little bit dense as well. You see, again, the graphical overview. You see that the axes are labeled. You see that some stars are added for statistical significance. That I have a bit my difficulty with, I explained already. But overall, things are labeled. I can understand what this is about. This is then the characterization of these particular cells in flow cytometry under which particular. Okay. And you may think that this is about the wealth of data being in the subfigures. No, it's not. You can also explain very well if you choose to do single panel figures. Here's one example for that I also liked. This is about a cloning experiment, and this person has depicted here details of the vector used, has stated then specifically here this promoter is inserted in this particular plasmid between these sites, and the agarose gel shows the bands of the backbone and this promoter of these sizes, and I can look whether that makes sense, and that's all that I want. I would have liked maybe to not only say this is a 1 kb gene marker, but really see the sizes here. Okay, but at least I understand what the point of this figure is. Now I see, I show you some more examples that I had more difficulties with. Again, this is no finger pointing, but yeah, maybe you can understand then how to make figures, how to make more complete figures. You see here a graph with an X and an Y axis that are not labeled. I don't know what is shown here. I don't know what is shown here. And I see a lot of colored lines that have cryptic abbreviations that I don't know what exactly they are. I see here that this is growth of a particular organism, and knowing that, okay, this is growth, I assume that this here is a timeline and that this here is an OD and optical density. I don't know it. I would have to go now into the written text and try to find it either in the methods, it's not in the legends, it's used I didn't put it in here, but this information is also not in the legends. I would have to go to the text and search for it, maybe even to the methods section and search for this text, what this exactly is. It's just easier and more intuitively to understand to have this information. I also know from the text that these here are all different clones of this organism in which single mutations have been done. But this information, again, could have been just simply stated here. Also, if I look at this, I wonder whether these three here, why these three mutants grow in a different manner. Is this significant? Are they followed up later? It's not. Another one, just showing one big electrophoresis. It's unclear what these ones are. This is DNA and RNA of some abbreviation. It is unclear here what does this person want to tell me. Is it good that I see these areas or not pointed out? Then it would be easier to understand. This one here is okay, relative good. I can tell you that I know these ones are particular viruses, variants of viruses. And here, this is, I guess, foci forming units, but it's not indicated per what. This experiment was done 144 hours post-infections, but the 144 hours post-infection does not belong to this axis. It's a description of the overall experiment. Here to this axis would have belonged that this is two different viruses with two different treatments, maybe, however you wanted to label it. What is missing as well is the information of these bars. Is this mean, medium, standard deviation, standard error? Is this, how many N is this? Is this significant? So I do see some differences, but are these differences statistically significant or not? Another one, this here is labeled, it's an mRNA expression level, but it's not said what it is and what is the unit. So is this, I don't know, I guess it's fold. And if I look around, I see this abbreviation here could possibly be a gene name, although nomenclature rules would state that a gene name should be in italics. But I assume this here is probably the gene name that we're talking about, and this here is the cell line we're talking about. And then, but it should have been here, that's the mRNA expression level of this gene in fold over control, or what it, or this would have been at least good. I mean, here it says then it's this CCL2 mRNA in the cell, but it's not immediately there. That's not my biggest critique of this, because I can get this information. The biggest critique here is that I have here stars for the, so this is samples of what? So what is the things you compare here? And you have indicated here the statistical significance with a star or 2 stars, meaning these p-values on an n is 2. And you cannot make statistics on an n is 2. N is not identified. And it is to me not clear whether this statistical significance is of these to this, or are these in between. So is here the difference between the normoxia versus hypoxia here as well, or here. So it's not intuitively well understandable. So yeah, I don't know if this here, so if the 2 stars should be statistically significant, which would mean p under 0.02, I don't see it with these error bars. I don't know. It's just difficult to really get the point. Okay. So to summarize the figures, Again, the results need a logical flow, etc. You define all the symbols and abbreviations. You define any sizes and bars and error bars, normalization, etc., number of replicates, and all these things. My personal tick in multi-panel figures is one font size for all labels. I fight over this with my people as well. It's just a personal tick, because already, if I talked about this earlier, if I already see these p-values here, I kind of have to go, what's the p-values there? They're tiny. And if you have one font size throughout the figure, there will not be one label where you that you cannot read, because it's all in one font size. Or you make the whole figure then so small that you can't read any of them. But that usually people don't do. So usually the thing that a lot of people do is that they make, for example, these A, B, C, they make those huge and huge other labels, and then a lot of other, they make these gradients of label sizes, and then there's often label sizes that are very difficult to read. Also something that I see in theses a lot, that something like these plots, Western blots, PCR plots, are, and also bars of bar graphs are huge, while the labels are then very small. So finding a star in one of these figures then, for example. And here it's just a friendly reminder that our eye can actually capture whether or not there's a big black blob here fairly well. So these sizes, they don't need to be that big. But we usually have difficulty in identifying these tiny symbols like a star. So try to, show your figures to other people and see whether they can read the stuff and follow this. For microscopy pictures, I usually like them rather big, because you want to see the point. This here is relatively small, but the major point that this figure shows here, that in this particular growth condition, we have no dots here. And in this, we have medium dots here. And in this, we have more dots here. So it's the amount of bacteria bound to these cells. And this, I think you can get even at this magnification. So compose your figure into making something that's intuitively understood. I hope this sentence that said each figure should stand for itself has now become a little bit clearer. So once you have now your figures, you start to write your story around this. And what I experience with this is often once you go through the figures, you actually see that things don't really flow that well as you thought they would. So then maybe you shift things again a little bit. There is a certain rhythm to the results section. And this rhythm goes like this. You state the problem, you state your hypothesis, your approach, your data, your summary, and this leads to the next hypothesis. Now, this sounds a lot like the overall paper, but I mean here that within the results section, every paragraph has this rhythm again. Every paragraph describing a particular figure or parts of a figure states again the hypothesis or what the overall problem was, what the gap of knowledge is, what you aim to do, and then how exactly you did this and your data, and a short statement of what this actually, that sort of is the main message of this data. Typical for this is to investigate this or that. We did this or that. Data showed so and so. This indicated that. Let's look at this in some examples. We take again this example paper and go to the section where we described this particular figure. It's here. To verify the preferential binding for Helicobacter pylori. That's what we wanted to do. To do this, we did this. Describe a bit more detail what exactly we did. And then showing what the data showed. And then the results show that 3 types of 2D monolayers are sort of our take-home message from this part that leads us to the next logical step in the results section. Let's look at some more examples for this. I just took a pretty random paper from a colleague of mine, simply because he's in the institute, and I thought for our students it makes a lot of sense to know a bit more the papers of the people that are there within the institute. But I would like to encourage you to take any other paper and look for this structure. I take here just the 2 first sentences. The last author is Juri Raptöber, and it's totally not my topic, but I just take the first 2 paragraphs from the results section. And I want to go with you through this again. Just look for the sentences. To test here, to test this or that, we analyze this or that. Describing the data, and here comes thus only, we conclude so and so. Okay, we can label this again. It's the same kind of structure. The next paragraph again. To do this, we did that. We could obtain this and that. Thus, the new protocol provided previously unavailable amounts of pure homogeneous and catalytically active yeast polymerized to, etc. Okay. This is again a matter of style, and there's other people that don't do it exactly like this. There's people that can write fantastic papers that are much more beautiful in the meaning of, and not that repetitive. I really follow this rule here quite strictly. I do try to find a bit different words every now and then, but I just follow this because I find it just very helpful to have a simple rule, and I find it very helpful to read in this way as well, because what I often do when I read papers is I go to a particular figure and I look at the figures and I try to understand the figures, and then if I miss some information there, I don't get exactly what this was done for and what this data means, and I jump to this section, and if I jump to this section, I see immediately, okay, to do this, we did that. This is the data and this is the main message. Again, it is very nicely digested for the reader, and I find that very helpful. But it's absolutely a matter of style, and this is certainly not a rule that everybody has to follow, and it's not written in stone, and there is other beautiful ways how to do this. Okay. Oh yeah, and probably now students may think now, okay, all this description is okay, and I can understand that a student will, and I remember that I felt like this when I wrote up my diploma thesis. It was back then, and I thought, all this you can write result sections of papers because there is actually so much data, and I only have so little, and I cannot break this up into single steps. It was really hard to digest this. I want to show you an example showing how you can break down things. So within this Helicobacter paper, there were actually several students involved here as well. And I will show you one example. So here in this results section, we describe here this supplementary figure 8. So to analyze the importance of MUC5AC and PSCA, 2 proteins, human proteins, we generated a CRISPR-Cas9 mediated knockout in human gastric organoids. Knockout was confirmed in DNA and protein analysis, but bacterial binding indicated that neither MUC5AC nor PSCA are required for Helicobacter adhesion to the 2D monolayers. Supplementary figure 8. Let's look at supplementary figure 8. This is the figure, and this here is the MUC5AC knockout. which has been done by one master thesis. And this here is the PSA knockout, which has been done on another master thesis. Now, these people obviously had to write down all these steps and had to break down all these steps similarly into these little results sections. I would take this one as an example. This here is now from the master thesis. And here you see the result in I obtained this particular knockout is broken down into a multi-panel figure. Into this here is my gRNA, my guide RNA that I use for the podcast. This is my plasmid, and I do a electroporation. Here grows my clone, and here I analyze 2 of the clones, and I see here that the PSA labeled here, PSA is less here than in the wild type control. So 1 point, just I made this knockout and I analyzed the knockout. How did this person describe this? Again, we can mark it with our colors, and it states here, this was the overall problem. Then to test the possibility of a causal relationship between PSA expression and Helicobacter attachment, I did this and that. This was a long description of the data that I shortened a bit, just to show you that below this, there comes taken together. This data suggests that this CRISPR-mediated genome editing of PSA led to an afunctional protein which should be localized to the cytoplasm and or quickly degraded after translation. Both clone 3 and 4 can be equally considered a PSA. So this is just a just one example of how smaller packages of data can be described in the very same, with the very same structure. And I hope you would find this helpful. Okay, so to summarize the results, you write your story around the figures, be open to shift things. You may realize that a particular statement cannot yet be done. And therefore, you may realize that some experiments are missing. You may need another N to do your statistical significance. So for this reason, it's actually good not to delay your writing of a thesis to the last minute when the lab work is already finished. It should be ideally written during your lab. Be concise and be objective, and there is a fine line between stating results and interpreting them. Interpretation belongs to the discussion. But this I find not that much a problem between in theses. So I think you know that you need to discriminate between what you see, and the data. Your description of the data belongs to the results section. The interpretation belongs to the discussion. Okay. Now, after you have written your results, it's a good idea to revisit your aim, because often people start out and aim to do something that they did not accomplish to do. A typical example would be you set out to make a knockout, but you just, for technical reasons, were not able to obtain this knockout. And this is not, especially in a bachelor or master's thesis, this is not a problem. You can still write your thesis. You just should not then write in your aim something that you didn't achieve to do. So after you have written your results, revisit your aims. Formulate your aims again to meet exactly what you did. How to do this? One example, in the example, thinking that you would look at a knockout, your original hypothesis may have been, I hypothesized that gene A may have a function for whatever B. To test this hypothesis, I will analyze a loss of function knockout. My specific objectives are generate the knockout, establish methods to analyze the knockout, and then analyze the knockout with these methods. Beautiful thesis, great. However, things went wrong and you didn't get that knockout. So you reformulate. I hypothesized that methods XYZ would be good for an analysis of function B of gene A. To test this hypothesis, I will establish these methods and test their usability. I will further start the generation of a knockout for the future analysis. My specific objectives are establish the methods to analyze the gene function, demonstrate the usability of these methods, and start the knockout for future analysis. This is clear that the actual generation of the knockout will be subject to future studies. So, but then if you formulate these aims, I do expect that you show me as well that you also demonstrate the usability of these methods. Okay? So state your aims precisely and in a way that you actually achieve these aims in your thesis. So the aims that you set out originally may not be the exact aims as they are stated in your thesis. Again, in the thesis, you tell a story. It's good to read. So in the discussion, you basically restate your original question, and then the main point here is now to answer this question. If you stated a hypothesis, pick up this hypothesis and answer this hypothesis. Have you solved it? What came out of it? Say that. Then explain why and how exactly your results support this particular answer. And you kind of put into this how this fits in with existing knowledge. So the discussion is not a discussion of why or not a particular experiment did not work out. It is technical details can be part of the discussion, but it should be a smaller percentage of it. The large chunk of the discussion should be to discuss and interpret your results in light of the current literature. And they should kind of embed your results within the body of knowledge that already exists. You can, in writing the discussion, you can differentiate a little bit how sure you are. So there's some good advice and videos about how to play with English language. There is a difference between you say this data suggests, or something implicates, or it can be speculated that it could be this or that. Future experiment would need to show this or that. I personally avoid novelty claims and avoid the name I prove this, or this data proves that, simply because you never know whether you really know all what is out there. So I don't like the novelty claims. Some journals also dislike novelty claims and actually filter them out every manuscript. And yeah, I just, we're scientists. We're trying to be careful and just simply state what the data is that we have, and what we think this data may mean, and what can be taken for this for the future. Questions to guide you during your writing. Why is this new? Has something similar been done before? What are similar findings? Where are discrepancies? Is anything in your study maybe very unexpected? And how could you explain this? Also really important, what are the limitations of your study? What are weaknesses, and how could future experiments maybe answer these weaknesses? Be critical. I like criticism. It's important. Why are your findings important, and what will be done building on this? In a thesis, and also in some paper formats, there can be a section conclusions and outlook, and especially this importance and what can be done that builds on this is part of this conclusions and outlook. So now that you've written your results, written all the beautiful story around it, you have discussed the data, you have shone on this data with the light of the current literature, and you know precisely your gap of knowledge and the aims, Now you know all the factors that you actually need to understand your story. Meaning now you know what you actually need for your introduction. So now these are the things that you need to introduce. They feed directly into your introduction. The introduction is like a funnel. It goes from very wide. So it's really kind of a one-way street that brings you directly to the gap of knowledge. And you pick up your reader with a general topic. Often this includes why this is important. And during this trip through the funnel, you have to introduce all these building blocks that the reader will need to understand your study. Think about your gap of knowledge again. What exactly are you addressing? This needs to be an important part. If your main message addresses this gap of knowledge, this is the topic. This needs to be in the heading, in the abstract, in the beginning of the introduction, in the gap of knowledge, and discussed at the beginning of the discussion again. So this is very strong. This needs to be super good. Why is your study important, and what did the literature know about this before your study? So this all belongs to the introduction. Sometimes in a thesis, you also pick up stuff that is done currently in the group, but not yet published. So this also belongs to the introduction. If you are now part of a particular running story that is not yet published, you need to give some background information why you have this particular gap of knowledge that you will fill. All right. Refrain from opening sidetracks that are not relevant. In the introduction, you tell me exactly what I need to know for this story. I don't need to know all the background stuff, what else is done in this department or group or whatever. I need to know what you need to know for this story only. So again, throw out stuff that you don't need to introduce. One example, well, of course, the Helicobacter paper. I hope you're not yet bored with it. So what do we need for our Helicobacter paper? Well, it's a Helicobacter paper. So we start with this. Helicobacter is really important because many people get sick. So we start here. In terms of prevalence, Helicobacter pylori is a very successful human pathogen that colonizes the stomach of about 50% of the global population. Okay? Now I need to tell you, because this paper is about the adherence of the bacteria to host cells, I have to tell you that is really important. Yeah. So Helicobacter evades the luminal gastric acid by replicating in the mucous layer close to the epithelial layer, or by colonizing the glands. Okay. So then I use many more lines to describe that the attachment to the epithelium is really important. Now, then I need to tell you, knowing that the attachment is so important, I need to tell you, well, there's so many different cells that the bacterium could actually attach to. Of course, they're all epithelium, but the epithelium consists of different types of cells, different types of epithelial cells. I have to tell you this. So I need to tell you, the gastric epithelium is organized into flask-like invaginations, the gastric units, which are further organized into the regions pit, isthmus, and gland. The pit harbors MUC5AC expressing pit cells, etc. So I really go down from this is the overall structure, and these are the different cells that are there. Okay. Now, I need to say that there is some evidence that Helicobacter may attach to specific cells, but, and now comes the knowledge gap, the actual target cell is elusive. This is the blocks that I need to tell you, and I tell exactly those blocks. I leave out all my interesting NF-kappa B stuff that I've also done, because it doesn't belong in this story. Okay. So in a thesis, if you want to put introductory figures into the introduction, I really like that. It's much appreciated because it helps me to grasp or to focus on important point, and visual, it just picks me up on a different level again and helps me to understand what you want to tell me. obviously, clearly show me the important points. And if you do this, remember to put a meaningful legend that also indicates whether this is your own creation or this is a drawing you added after inspiration from somewhere else. Where is this? Or whether this is taken directly from a specific source, like a paper, then this has to be referenced. In the material and methods, the goal is here really to provide enough experimental detail so that anyone in the world can repeat your experiments. It goes from an organism studied, a cell line, a culture technique, etc., to an experimental design. Then you explain the data collection, data analysis, and then the statistical analysis. You identify material precisely, name some brands. This is, again, comes with practice. Which information do we need? I usually don't need the information which brand a particular centrifuge comes from. But if you talk about antibodies that are super specific and give different kinds of stainings, I need to know the brand and even the catalog number. I do need to know the sequences for primers, everything that you really need to replicate your exact findings. Provide enough experimental detail. Again, this is hard to define. Look at papers. They're usually very, very short, much too short. And look in comparison to maybe an SOP or a standard operational procedure, or in a longer experimental detail that you receive from colleagues or methods papers. So there's a huge bandwidth, and I'm usually fine with a rather concise version of it. But this is up to you in a thesis, for my taste. I want to know what is concentration, etc. You want to see it is centrifuge at so much G for so many minutes at which temperature. Don't say RPM, because the RPM is specific to the centrifuge. Then I need the information on the centrifuge. To avoid that, just name the G. That tells me just as much. Tell me molarities, not whatever dilution. And here's the nomenclature rules that I named beforehand as well. So with this, we are at the abstract. For the abstract, there is a really nice example from Nature, and I've just taken this from Nature. There's other papers that have it rather structured, and they really give you also within the abstract that you have to write an introduction, a methods, results, and conclusion. For example, gastroenterology or gut, do it that way. But normally, and in a thesis, I expect one flow of abstract. The abstract is really important. It really summarizes your whole paper, your whole scientific achievements, and narrows them down, boils them down to sort of the show phase, to what the reader will first see, and invite them to read the. So there is what Nature provides here is, again, the specific flow of how many sentences you put on which detail. Let's go through this. You put about 2 sentences on a basic introduction. You add some more details on this. And again, you have to state the general problem, the gap of knowledge. Then, following from the gap of knowledge, you state the main result. Here we show. Then you provide a bit more detail, exactly what you found. Put this into a general context and draw some broader implications. Here's some example of one paper that Nature showed here as an example. This is about microtubuli, and you start with just cell division, very, very broad. And there's particular hypotheses, and then you come to the gap of knowledge. However, the precise roles of kinesin 5 during this process are unknown. Now comes the main result. Here we show that so and so. Then some more detailed results. And now these results are put into a general context and even more broader implications. The broader implications mean something like we anticipate that our assay would be a starting point for more sophisticated in vitro models. Or EG5 inhibition is a major target of anti-cancer drug development, and a well-defined and quantitative assay for motor function will be relevant for such developments. Why is your study important? And I have to confess that especially this last part, this is another example. Again, some basic introduction, some more background details, yet remarkably little is known about lipid droplet cell biology. So again, very clearly the problem, your gap of knowledge. Here we show what is your main finding, detail this a bit more. And this person also doesn't like so much more of this broader context, but reduces this to one statement of what's the broader context of results. Okay, now one example from the Helicobacter paper. Yeah, put the colors into this, and what did I do? Well, I stated the overall introduction. I state the gap of knowledge, but the possible host cell tropism of Helicobacter pylori is currently unknown. Our main result, here we report that Helicobacter preferentially attaches to differentiated cells in the pit region of gastric units. Show a bit more detail, and then I kind of escape and don't really want to use the overall conclusion or the broader implication of what we find. I think this year we had to stay within a size, a word count limit, and it's really hard to then boil it down to match exactly this. And somehow this last bit got lost. I'm not perfect. Other people do this better, and I look around in colleagues' papers. Again, this here is this is from another colleague here at the Institute, Jens Kurik, about RNAs, and he did this very nicely. Here, the overall problem. Here specifically, it's about these G quadruplexes. Their overall biological relevance remains elusive. Here we demonstrate, novelty claim, okay, that an RNA G quadruplex structure inhibits translation in vivo in eukaryotic cells. More details and an implication. Very nicely. Another example, we take the paper of Yuri Rapsilva that we also had for the results section, and this is also very nicely done. Main problem, gap of knowledge. Here we show, state your main results, some more details, and then comes the conclusion. These results suggests and a broader implication. Now, let's go to some practical advice that does not belong only to one specific section. The tenses are a topic in themselves. I have put a very nice overview in the next page. The summary of this here is in the introduction. You use two tenses, present tense and past tense. Past tense for stuff that people have done, but present tense you can also use for general statements like we all know that the world is round. Okay, I don't know, something very general that is accepted, you put in present tense. The methods are past tense to describe what was done. The results are past tense for results obtained. Present tense if you point to specific figure, like figure so and so shows. If you point to something that is seen right there, because it's not. In the discussion, you use the present to explain the significance of results, and past to summarize specific results. It's good to read this a bit more in detail and think again on how you can use this. I would like to highlight this. This is from the University of Melbourne, just a 2-pager on using tenses in scientific writing. I like this a lot, and this actually hangs in our writing office for many years now. I find this very helpful. So you can Google it, or for our students, I put the PDF for you in the course. Okay, in a paper, often to use the active voice, we use we do this, we do that, here we show. In a thesis is showing your work and your work only. And you actually sign a statement that what you show here is your work. So we is kind of difficult to put in a thesis. You should say I, or find some work. With this, we are also I have to mention plagiarism. Avoid the use of words or ideas or images without citation. Be precise in what you did and what others did. For example, if you say you used a particular method that may be more complicated and you used somebody's help to do this, I don't know, doing electron microscopy, you may have prepared the sample and you may have accompanied somebody with the whole process, but not did this all alone. And you state this in the figure legend where you show these images of the electron microscopy and name it in the method. Reference everything that you used. Now we come to this part of practical advice, when to do what. This is something that I find very important because I did the same thing, putting everything to the last minute, and then this writing just becomes a huge mountain of never to climb hill. So I try to break this down for students that do their thesis within my lab. And I learned this also from actually students that came to me and told me from experiences in other labs, and I like that very much. So obviously what you do first, you find a lab for your thesis. Clear. Okay. Within that lab, they usually have some sort of project running, and you get the possibility to take part in this project, and they define an aim. This you should already take part in, define a particular task that you enjoy doing. Now you need to really sharpen your hypothesis and aims. And if I am asked to be a university supervisor, I jump in here and I really want it written down. What is your hypothesis and what is your aim? Simply to help you sharpen this at the beginning. So when you start your project, you know at least what you aim for and what you envisage to start. Now, with this, your thesis can start. After some rounds of discussion, obviously, you need to sharpen your argument. But after you did this and supervisors are good with this, you can start. If you would do your thesis in my lab, I would ask you to now write within the first week, to sit down. You did already some literature research because we had those discussions and we discussed your aim. Now that you're in this, sit down 1 week writing this, and within 1 week, you just send me by email a Word document with 2, 3, 4 pages, whatever you like writing, of your introduction. Sounds hard. It's just a preliminary introduction. You're not evaluated based on this. This is only to help you defining what is known in this, because this makes you sit down in the beginning, put something on paper, and only putting on paper makes you actually write the words and know exactly what you do. And just sending to me just helps you to actually do this. So Exactly. After this, you go to the lab and you do all your fantastic lab work. And while you do this, you always need to write down your methods anyway. So why not write the methods in a kind of style that you can also use for your thesis? And while you're also in the lab, you need to discuss your data. And I would strongly encourage you to also present your data in the lab seminar of the lab. So you make some figures and you present them, just 2 or 3 figures, because you need some time to generate some data. So you may not have that much data, but just even if you have a single figure or 2 figures, just put them in a PowerPoint and present them to the group. Get some feedback, hear whether they understand that. And if you then make your statement, this statement can be the heading of the PowerPoint presentation. This helps you to already test and sharpen your statements, and it makes you realize if you yet cannot draw a particular conclusion, making you go back to the lab work, producing that last N you need, or producing the results again with the correct controls, etc. Okay, so this you should do already while you are in the lab. Obviously, with these iterations that you need to discuss your data, to make the figures, and to sharpen your experiments again. Now, once you've done that, you write this story around your figures. You optimize them, and then obviously this takes time, of course, it's a lot of work, yes, but at least the page is not empty and you have something to work on. Now, after you have then written your results, you just do the discussion of these results, because now you know what you need to discuss. This will take time. This is super hard work. This is a tough region to write. But then once you have written this, you need to revisit your aims, as we discussed. Did you really do? And then you visit that introduction again and sharpen that introduction, and you can discard parts of it and rewrite parts of it. It's to fit really the project that you did in the end, and to come down to this gap of knowledge that you actually managed to answer in the end. You need to do the abstract, get lots of feedback on this, show it to colleagues that work in your lab, show it to other people that also write their thesis. Do they understand your stuff or not? Show it to their supervisor, get some feedback. And then, yeah, see, the whole writing process actually wasn't that bad. I hope. So you're there, you have written your thesis, and I look forward to receiving it. And I hope that this is helpful. It works for the people in my lab, I hear. So some take-home messages, I actually got those. So this here is this using tenses in scientific writing, this piece of paper that is hanging in our writing office, in the PhD office. And I scribbled here these notes at the sides, and this is kind of the scribbles in nice. So the order of writing: your first week, write the intro and aim, ongoing, results and methods. When results are done, write discussion, last, refine intro to really introduce what you need, and only that. The results, subheadings are sentences stating results. The figure legends, I like them as statements as well, but this is a matter of choice. Each figure needs to be understandable alone. I hope by now you understand what this means. The discussion is an embedding into literature, something again about units. There's a space before a unit. It's, for example, 10 space micromolar, use G, not RPM, etc. Nomenclature. Then this is my version of these sentences rules for the abstract. Just some sentences background. The main aim needs to be in here. Sentences for the methods, sentences for the results, and some conclusion. the tenses are best to once read in this example from the university. All right, so with this, I would like to end. I hope this was helpful. Yeah, I would appreciate feedback. As always, this is helpful. Maybe whether you need another version of this lecture, or if you need more help. But I hope that this was helpful, and I wish you all the best for writing your thesis and your future papers as well. Or if you're at a later stage and already beyond your thesis, I hope that you write many exciting papers and that this was still helpful for you. Thank you very much. Bye-bye.